little bit more simplified diagram of that Hershey and Chase experiment we talked about yesterday. So they did 2 rounds of this experiment. In the first round, they used radioactive sulfur, and so that labeled the protein portion of the virus, right? And in the other half of the viruses, they used radioactive phosphorus, which tagged the DNA. They let the viruses infect the bacteria. They blooded them to separate the extra virus parts from the bacteria, and then so when they were done, the protein did not end up in the cells, but the DNA did end up in the cells. So yeah, that just goes to show that it's the DNA that's getting passed on that's transferring that genetic information. Right? So hopefully you're able to do really well in that 8.1 quiz last night. If you didn't, you can still keep taking it, but I forgot to check to see how you're doing on all that. So hopefully you did okay. Today, we already finished part A, so we're going to talk about part B, which is basically looking at the structure of DNA. So we're going to locate label parts of a nucleotide and look at a DNA model and also look at the base pairing rules. So if you open up your folder to the next blank page, I haven't worked on yet, which is chapter 8, part 1, page 4. We started actually talking about this a little bit yesterday. DNA is what type of macromolecule? Nucleic acid. Remember what the monomer of nucleic acid is? Nucleotide. So DNA is only one type of nucleic acid. So, I didn't mean to have those up there yet, but there's a picture there that should look just like this. So what we're looking at is 3 parts that every nucleotide has. So the little circular part here, that's called a phosphate group. So the phos part comes from this P, which stands for phosphorus, and that phosphorus is bonded to 4 oxygens. And this might be important if you go further into biology, but those oxygens are negatively charged, so that's going to affect the way that they interact with other molecules. If you're negatively charged, you're going to be attracted to positive charges and repelled from other negative charges. In the middle of the nucleotide is a sugar, a monosaccharide ring. So I mentioned DNA is only one type of nucleic acid. So in DNA, that sugar is deoxyribose. That's where the name DNA comes from, deoxyribonucleic acid. RNA, which we'll look at in the second part of this chapter, has the sugar ribose. So that's where its name comes from, ribonucleic acid. And the third part of that nucleotide is our base. So sometimes we just say base, but it's a nitrogenous base because it contains nitrogen. The nitrogen's in there. Usually we just say base though when we're talking about those, just because it's easier to say. So every nucleotide that you look at in DNA or RNA is going to have the same structure. It's going to have a phosphate connected to a sugar, connected to a base. There are actually 4 different bases, so those letters probably look familiar. The C stands for adenosine. The T stands for thymine. And the G stands for guanine. And the A stands for adenine. So, in this particular diagram, cytosine and thymine are together because they're fit into what we call the category of pyrimidines. Guanine and adenine are together because they're both purines. So in your groups, what do you think the difference is between these pyrimidines and these purines? Look at those structures. Yeah, what's similar about cytosine and thymine compared to guanine and adenine? I heard a couple people say it. These are made up of one ring, right? And these are made up of two rings. When we talked about base pairing and those ratios, Chargaff's ratios yesterday, we know that C and G come in equal amounts, so they always pair together. And A and T come in equal amounts, they always pair together. So we always have a pyrimidine paired with a purine. So when we pair those up, they're going to take up the same amount of space because we have a double ring with a single ring and a double ring with a single ring. So, on the next page, there's a couple of scientists we're going to talk about today that help to determine the structure of a DNA molecule. Rosalind Franklin is the first one we're going to talk about, and she specialized in this process called X-ray crystallography. And so you probably would have to take a whole class to really understand how to read these. But the importance of this was that from this picture, if you know how to read these pictures, we can tell that DNA is a double helix. So we know that from this picture that it's made up of two strands and that those strands are twisted around each other. And probably most of you are somewhat familiar with that DNA shape, right? It looks like a ladder, but twisted. So that's what a double helix is. You have the two sides of the ladder and they're twisted around each other. Watson and Crick, however, were the guys that really got credit for coming up with the structure of DNA, even though they used Franklin's work without her permission. So from Chargaff, they learned that A equals T and C equals G. And from Franklin, they learned that DNA was a double helix. And that and another couple of things were able to help them to put together this final structure. That the bases are in the middle in those pairs, A and T and C and G, and that the sugar phosphate portion of the nucleotides make up those sides of the ladder. On the next couple pages, there's some questions that I want you to take some time to work through with your group. So there's a Yeah, so you're gonna use these pictures to answer the question. So yeah, go ahead and get started.
Go for it.